Standard PCR reaction. For preparing a standard PCR reaction of 50 microliter, if we see what are the main ingredients which are required to perform a PCR reaction, they can be 0.25 millimolar of each primer, 0.2 millimolar each of the DNTPs. There are 4 types of the trinucleotide phosphates: adenine trinucleotide phosphate, cytosine, guanine, and thymine. So 0.2 millimolar each of the DNTPs, 50 millimolar of KCl, 10 millimolar of tris, which is usually used as a buffer, 1.5 millimolar magnesium chloride, 2.5 units of the DNA polymerase, and 10 raised power 2 to 10 raised power 5 copies of the template. So these are the main ingredients and their amount which is required to perform a standard reaction of the PCR. The volume of the PCR reaction can be 50 microliter. But this is a standard reaction. There can be variation in each of these ingredients because sometimes we have to increase the amount of the DNA polymerase. Sometimes we have to increase the amount of the magnesium chloride. So this is the requirement of a standard reaction, but it depends on the primers, it depends on the amount of the buffer and amount of the magnesium chloride, because before optimization of the condition, sometimes we have to change or vary the amounts that is given here. Once all these ingredients they are mixed, then there is the PCR temperatures. Because PCR consists of 3 steps. The first step is denaturation, second step is annealing, and the third step is extension. So during denaturation, double stranded DNA is converted into single stranded. That can be between 90 to 96 degrees centigrade. The next step is annealing temperature that controls the specificity of hybridization, mean the joining of the primers with the template DNA. That can be between 40 degrees centigrade to 68 degrees centigrade. And the third step is extension temperature, usually that is 72 degrees centigrade, but there can be variation between 72 to 75 degrees centigrade. So these are 3 different types of the temperatures. Denaturation temperature is different, annealing temperature can be different, and the extension temperature is different from the other ones. So if we do a comparison between different temperatures and what kind of product we want to obtain as a result of a standard PCR reaction or as a result of a typical PCR reaction, here we can see if the temperature can be kept 94, 60 and 72, that is perfect for long primers. So higher temperature can be used, maximum annealing temperature. Second type can be 94, 55, 72. This is good for perfectly matched primers. So this is usually considered as standard condition of the PCR. Next type can be 94, 50, 72. That is for adequate primers. In such type of primers, if there are 1 to 3 mismatches per 20 nucleotides of the primer, if there is some mismatches between the primer and the template, then this type of the PCR prom is used. Next type of the PCR prom is that can be 94, 48, 64. That is usually entered into the PCR machine for poorly matched primers. there can be 4 to 5 mismatch per 20 nucleotides, and the last can be 94, 45, 65. That is for unknown match between the primer and the template DNA. This kind is used for primers of questionable quality. So these are some of the common temperatures which are used during the amplification of the DNA by considering what kind of reaction is being performed. Let's say if there is long primers, then the first option is adopted. And if there are some mismatches between the primers and the DNA, then we can use these kind of the proms in the PCR cycle. It depends on your requirement, and it depends what kind of amplification you are going to do. So temperatures, they are actually used for denaturation, for annealing, for extension. So during the PCR cycle, temperatures used for denaturation, for annealing, and for extension. Amplification prom usually varies between 20 to 50 PCR cycles. During a standard PCR cycle, 30 to 40 cycles are sufficient, but in some type of the PCR, even 50 cycles are required to perform a complete PCR reaction.